Good evening, everyone. Thank you for joining us for tonight's Alumni University presentation. We are so happy to welcome Phil Troyk. Before we get started, just a couple of housekeeping notes. Just a reminder that this is a webinar. So if you have any questions throughout the presentation, please feel free to include them in the chat box. This webinar is also being recorded and will be available on our website later this week. If you are having issues with your audio or video connecting, it could be your own internet connection. So we encourage you to close out of Zoom and reopen it. So to introduce tonight's presenter, Dr. Phil Troyk is the Executive Director of the Pritzker Institute of Biomedical Science and Engineering, as well as a Professor of Biomedical Engineering and an Affiliate Professor at the Stewart School of Business here at Illinois Tech. He's also a faculty associate in the Division of Biological Sciences at the University of Chicago. He holds a bachelor's degree in electrical engineering from the University of Illinois, an MS in bioengineering from UIC, and a Ph.D. from UIC as well. So with that, I will turn it over to Phil to begin tonight's presentation.
Thank you. And Kristen, I just had a question about the about questions that might appear in the chat. So
Sure.
Should I be trying to look for those as they come along or no?
I will be here monitoring for them, as well as my colleague Joe Volan.
Great.
Will be watching for those as well.
Okay, thanks. I appreciate that. Okay, well, thank you everyone for being here and coming to listen about the research work I do. It's always a pleasure for me to tell people about it. And I've certainly been blessed to be working in something that I can get excited about every single day. So I started in electrical engineering and was one of those electrical engineers who got converted to bioengineering and biomedical engineering because it just fascinated me to think about how I could take electronics and to mate it with the nervous system in the body. And so after a somewhat long road from transitioning from electrical to bioengineering as a graduate student, first at Urbana in electrical and then here in Chicago at UIC for bioengineering, the Pritzker Institute was just being formed by an endowment from Bob Pritzker. And so I came to IIT and didn't plan to spend most, if not all of my career here, but it just sort of worked out that way. And it's been a wonderful place to be at. So without any delay, I would like to just start the presentation and tell you about the work that we have been engaged in. For a number of years, and I'll get into that in terms of the history. But for us at IIT, it has been a 20-year quest to come to the point that we are. But certainly the project has histories that are even deeper than that, and I'll tell you a little bit about that today. So this is addressing an issue of blindness, and it's curious to think about what the impact on our society is of blindness, because it isn't something that a lot of us perhaps think about. We take sight quite for granted, but if you use the every so minutes parameter, and these are slightly dated from the World Health Organization, but every 7 minutes, somebody in America becomes legally blind or is severely visually impaired. And currently, it's probably closer now to 15 million that are affected here, and 1.6 million are completely blind. And there's been a progression of increasing blindness in seniors, but it also it fits age children, school-age children also. And the cost of it is pretty dramatic. 70% of working age people with blindness are unemployed. The annual cost to the economy, even 2-3 years ago was 4 billion. But on a personal level, not being able to have sight produces social isolation, depression, disconnection from others. And I like to tell people in presentations when I can see them face to face, but I'll mention to you now, just try an experiment. Close your eyes and just spend the entire time of the rest of this presentation with your eyes closed. I'll bet that after a minute or so of doing it, it'll become irresistible to open your eyes. And that's because almost half of our brain is dedicated to vision. Blindness worldwide is a serious problem. And in the Americas, it's just a small percentage of the prevalence worldwide. But this is a staggering statistic from the World Health Organization that in what one might call developing countries, the expected lifetime of a child with blindness is less than 1 year. And it's uncertain whether that is due to social incapacity to deal with it, or whether it simply has to do with a breakdown of the infrastructure. So it's a serious issue, and many people in my field have been dedicated to applying technology to do something about it. And this is an artist's rendition of our system that you'll see a couple of times throughout this presentation. But basically, it's using technology to intervene in the brain to try to take images and put them into perception in the brain. So this is a little diagram of the vision system, and there's different places that one could intercede to put artificial vision information. The first obvious place would be the optic nerve. And there are such devices that go in the eye, in the eyeball, on or behind the optic nerve. And the idea here is that an implant that would be located there applies electrical stimulation to what remains of the neuronal structure in the retina. And by stimulating a certain location on the retina with a certain geometry, you can then basically produce an outline of an image that is captured by a camera. So there have been a number of retinal approaches. The next idea one might have is to go to the optic nerve. And there have been approaches for that, in which stimulation devices have been placed directly on the optic nerve in order to look at how one might introduce artificial information. In our case, we're going to the brain, where you have projections of the optic nerve that go through the center of the brain in what's called the thalamus to what's called the primary vision cortex. So this is kind of the concept where it always starts. The idea that you'd have what's called the scoreboard concept. And some of you may remember a ballpark scoreboard that used to be made of light bulbs. And with that, so many light bulbs, you could form an image. Or maybe you remember a light bright toy, which was these little colored pegs you would put into a lighted box and you'd make an outline of an image. Now, it turns out this is an extremely naive concept, but it remained the concept for quite a number of years. And the thought was that if you started with a face, you might be able to produce something shown here. We'd probably be lucky to do this. So when you stimulate in the vision system, you get the perception of a spot of light, and that spot of light is called a phosphine. And the idea is that with enough spots of light, you could actually get some type of an image perception. So there's a number of these projects worldwide that are being produced, not only ours, and they all use some form of interface at different portions of the visual system in order to try to introduce this artificial vision information. So in general, in this field, it dates back in at least for somewhat modern technology to the late 1960s. And at that point, people involved in the field knew what we wanted to do. And by the late 1980s, we had a knowledge base, but we didn't have the technology. From about 2000 to 2010, we, and I say we in particular at IIT for the project we're talking about, developed the fundamental technology base. And more recently, we matured it, and now we're into pursuing a clinical trial. So this is an interesting historical note here. This is a radio electronics issue from 1958, in which, based upon a patent by Shaw in Iowa, there was an experiment done. And notice the tagline here is, "Will the blind see by electronics?" And here's the device, 1958. It had clips to go to brain electrodes that actually went through a hole in the skull. It had photocells that the person could sort of move around the room and look for things like candles on a birthday cake or lights in the room. It had a battery, it had a variable oscillator and knobs the person could turn. And it had all the elements of a modern vision prosthesis system. And it cost $9.45. So I'd appreciate that none of you mentioned that to the NIH, because the NIH is funding us for $12 million. So an interesting historical development is that in 1966, the first conference on cortical vision prosthesis was held at MIT. And the conclusions were, you won't produce stable phosphines, the electrode material would poison the brain, and stimulation threshold would rise until you couldn't deliver current safely. Then in 1968, Giles Brindley in the United Kingdom, which I'll tell you about a little bit more, revealed his experiments, which disproved all of those conclusions. And in 1968 started what became the National Institutes of Neurological Diseases and Blindness, which then became National Institutes of Neurological Disorders and Stroke, in which it was recognized the importance of something like a neural stimulation system for vision prosthesis, at which then in 1969, there was a second conference on cortical visual prosthesis held right at the University of Chicago. And there's even a published book on it. And those conclusions were that the cortical vision prosthesis were not only feasible, but they were likely to restore useful vision perception. So it just shows how one development in the field of science can take the entire conclusions of the experts in the field and turn it upside down. Our project actually has its beginnings back to 1968, when the NIH formed a neuroprosthesis program. And it progressed forward from there, each having different stages of development. And in 1996, we at IIT, with me as PI, received a contract from the NIH to develop implantable electronics. The NIH approach to do that was somewhat misguided. But around 2000, the NIH canceled their intramural project and it became an extramural project, which then we wrote several proposals for, and we formed the ICVV project that I'm telling you about today. My own personal involvement spans from about 1984 until the present. So I've been involved with a project like this for a long time. So the intracortical approach is really one, in our case, in which we are taking modules that contain microelectrodes that get inserted in the brain. And these microelectrodes, if you were to compare them to the size of a human hair, there's something like this. So you, on the tip of a human hair, you could probably put 10 of these electrodes on the tip. That's how tiny they are. And in this module, which then has electronics and wireless communication, you can call up any electrode, and by telling the electrodes what to do and how to stimulate, the idea is that you could create the perception of an image captured by a camera in the person's brain. So if you compare these to other kinds of neural implant devices, cochlear implants for hearing have maybe up to 22 channels. Stimulation for paralyzed muscle, maybe 25, maybe 50. Heart pacers or diaphragm pacers, 2 to 4 channels. To even get started in vision prostheses, you probably need at least 500, maybe 1,000 channels. So the conclusion is that we had to make a system that was 5 to 10 times more dense physically and electrically than anything that previously existed. And that still remains true today. This is the seminal work that was done in 1968 by Giles Brindley, and these are remarkable x-rays. This is the x-ray of a person's skull, and you can see here these electronic packages, each the size of a quarter, an individual package for each electrode that was placed on the brain. And there were several versions of these, and one person actually had 3 of them. And I knew the bioengineer who designed this. His name was Peter Donaldson. And here's Giles Brindley and the recipient of it. And here's another version of the device. And you see the electrodes that are kind of wrapped around a portion of the brain here. And then they would map where in the vision space these little spots of light called phosphines were produced. And this was the last version that this person had in 1982. And you can see here, there's an individual transmitter in this bonnet that she has for each of the devices that were in the brain. This work was carried on by others, and they eliminated the electronics and they put a connector on the side of the head. And a version of that still remains available today in many experiments for brain-machine interfaces. Here you can see the electrodes implanted, and you can see a cable, and these are the dots in the vision field that show there. We actually have the brain of this individual, and we've done some pretty remarkable histology on it after he had been implanted for 34 years. So all of these prior systems had electrodes that were put on the surface of the brain. And now we're looking at what are called intracortical, which means that the electrodes penetrate the brain and go down to a layer that receives the input from the optic nerve. So basically half of your brain is dedicated to processing vision in one form or another. And that's a lot of neural machinery. And we're attempting to put in this interface so we can tap into and manipulate that neural machinery in order to try to produce the perception of vision. So the early devices, the early electrodes that were made with these were really quite crude. They had a little epoxy ball, and it held a couple of these electrodes, and it had wires that left off, and these would just be inserted into the brain by hand. And then it was soon recognized that one needed a better way. And so arrays of electrodes were devised. And these are some of the very early ones which look like a miniature hairbrush that had these electrodes sticking out. And the thought was then you could insert groups of these electrodes all at once. These electrodes are highly engineered. The tips of them are sculpted to certain shapes, and only the very tip of them are exposed, and the rest has insulation. So this is a non-human primate experiment we did in 2004. And here you see some of these arrays sitting here, 16 electrodes each. And what you should notice here are these cables that are coming from. Now, these are actually quite small. They're only about 1 millimeter in size, but this was only for 96 electrodes. And so if you imagine how many wires and cables you'd have to have for 1,000 electrodes, it just didn't seem surgically practical. And so recognizing that in all the animal work that we did, maybe 100 electrodes were used, and we're talking 10 times that amount. So the conclusions were that the cables had to be a lot smaller. They had to go away. And so we chose to make the cables go away. And then we envisioned what many people said could not be done, which was a miniaturized wireless floating microelectrode array, each module having 16 electrode drivers, each one being randomly addressable. And here was a concept drawing of that. And it shows the electrodes, it shows the electronics, it shows a coil for power and telemetry, and an interconnect area. So these are actually quite complicated little devices. The electronics, you don't order them from what used to be Radio Shack, or you don't order them from DigiKey or any other place. We have our own custom electronic chip that we designed for that. The electrodes remain highly sculpted, and they have to be packaged, the whole assembly, in such a way that makes it resistant to body fluids. And here you see a picture of one of them from the side. Now, these devices operate through what's called magnetic coupling because they're way too small to use batteries. And the battery technology is not available to have the power that would be required. And so we create an intense magnetic field outside of the scalp, and then we couple magnetically to the device through the coil that's there. And then the implant can actually talk back to us through the same magnetic link, so we can have two-way communication. And here you can see, once again, this is a pretty complicated little device, and we've spent quite a bit of time engineering it, and it's packaged in such a way with a polymer in order to protect it against body fluids. Here they are. Here's what they look like, and you can see one next to a penny. You can see the electronics in the interconnection. You can see the coil of wire for power and communication, and you can see the micro-sized electrodes that are there. So the first thing one might ask is, how do you surgically implant these? Well, for most of these types of devices, you have a cable that comes off. And so you hold it by the cable like a little handle, and then you can actually push it in or implant it. Well, that's completely impractical because we have no cable. And there would be no way of handling these things, even for a skilled surgeon, without damaging them. And so we developed the idea of putting them in a protective cylinder called a collet. And when the device is in a collet, then it can be loaded into an automatic insertion tool that I'll show you more in a moment. And this is a cadaver. And here you can see 9 devices, and these were easily implanted in less than 10 minutes. So here's a more of a modern version. These are little plastic collets, and they hold the devices in these collets. And now this gives a way to be able to handle these without damaging the electronics or the electrodes. And in order to even handle them, we have a specialized tool that we also developed that has like a little silicone rubber boot in it. And this holds the collet, and the person is able to manipulate it. And you'll see here in this animation, you see here's this little tool. Here is the little boot that holds it. This is a special insertion tool that actually shoots them in to the brain, and that's the way you minimize tissue damage, just like a nail gun. And so the device in its little protective collet is screwed into this insertion tool. The insertion tool is then placed over the brain, and then this plunger is pushed out like a tiny nail gun. The plunger retracts, and then one is ready to take out the little collet and put in the next device. So that was an animation. Here's a real one in the laboratory. And you can see a laboratory technician here screwing in and unscrewing the collet from this, which is this high-speed insertion tool. And here you see it being done the first time in a surgery. This is the first time that a surgeon is handling this in a non-human surgery. But you can see the surgeon screwing it in. And this inserter tool nail gun actually shoots these into the brain at a very high speed. It's about 1 meter per second. And the idea there is that just like a nail with a nail gun can shoot it into a board or a structure, these get shot in, and they actually minimize the tissue damage when that happens because they go in at such high speeds. So here you can see the surgeon taking the tool and putting it over the surgical field, and is now positioning it on the brain. And there you can see there's one device in already. And here, if you watch carefully, you'll see just a momentary motion here as there it went in right there. And there's the device that was just shot into the brain. And so they're very easy to put in, and they can conform to the brain naturally. And because there's no cable that connects them, it minimizes tissue damage. So here you see closely, here's the tool, here's the collet, here's this little plunger that comes and pushes it out at high speeds. And this is in a non-human primate. And you can see here, here's the devices right on the surface of the brain. And these go right in through blood vessels. And they don't cause any bleeding because the electrodes are so small, and they penetrate, and it immediately seals right around the vessel. So this is this same implantation. And these are some very remarkable MRIs, which is a 9 Tesla MRI. Typical clinical ones are 3 Tesla MRIs. And that was after 1.5 years of implant. And you can actually see the shadow of the devices here. In a normal MRI, you can't see them, they're invisible. But this is an interesting view here that's on the left, the rotating view, because this is actually the MRI of the brain of the animal. And you can see there's these 2 kind of depressions here where our devices were implanted. And on the other side of the brain, there were wired devices implanted. And you can see these giant cavities that are here in the skull from the constant motion of the devices. And here you can see another blow up here of the devices barely in shadow that were in the brain. And this dimension, by the way, is only 2 millimeters, this whole dimension right here. So perhaps this is something interesting to look right before dinner, but this is the histology, what the brain looked like. And this is after 1.5 years. And there's just a very, very mild layer of tissue on here. And remember here that the difference between this and possibly having these cables, and you get this because the device is totally floating and completely wireless. And you can see there's just a minor depression that was here. And scientists and life science people analyze this by what's called histology. And so what this is a staining of the cells of the brain after the device was removed. And what I'll point out to you here is that these holes are where the electrodes are. And you don't see around the electrodes any depletion of cells in the brain. And if we stain it with something that looks for neurons, which is these brownish things, you don't see that there is any damage that's being done or any loss of cells. In fact, for some electrodes, the neurons are right on top of the electrodes. So this is a really remarkable result and is not seen by any other devices. And we feel this is attributable to the fact that the cable is gone. So the cable not only has the benefit of removing the possible pathways for infection through the dura barrier of the brain, but it also seems like they allow the devices to float with the brain and prevent damage. Now, the electrodes are made of something that's called activated iridium oxide film. And what that really is, for those of you perhaps from chemical engineering, this is an oxide film that's grown on the iridium, and it produces very special effects for being able to inject current into the tissue. This is one of these electrodes. This is 20 microns. And remember, a hair is about 100 microns. So a hair would be 5 times this big. And this portion here is the insulation of the electrode. This tip is where it stimulates. And these films are actually what's called electrochromic, which means that they can change color depending upon the stimulation. So you may be wondering why this is called Goldilocks. It's because in the early stages of our work, we had quite a debate in our team about whether an electrode should be too sharp or too blunt, which should be too large or too small. So coming off of the old fairy tale of something that was too small, too big, or just right, we decided we would call our electrodes the Goldilocks electrodes. And again, this would be the size of an electrode compared to a human hair. Now, if you're a watcher of public television or Discovery Channel, you may remember where iridium comes from, and it's found in the KT boundary of the Earth's crust. And that material is there when the asteroid that hit the Earth, that is theorized to have wiped out the dinosaurs, threw up into the atmosphere iridium that is found in this one KT boundary of the Earth's crust. So we like to say that we're using material from outer space to make our implantable devices. Unfortunately, 90% of the life on the planet Earth had to get wiped out so we could get it. And so looking at this device a little more carefully, and in particular the electronics that are there, we designed all the electronics ourselves. And this is a CAD drawing of what a chip looks like. This is about 2 millimeters on the side here. And each one of these tiny little dots, you can't even see our transistors. And there's 16 identical looking segments all on the outside. And these are really the drivers. These are the things that talk to the electrodes. And then we've got logic in here that listens to commands coming in, and circuitry that process power. Now, most devices that are implanted look like this. These are titanium cans with wires that come off the top. Almost all implantable electronic devices look something like this. And when they're put into the head, this is what they look like. And our devices are obviously a fraction of this size. And we plan to put about maybe up to 40 of these in the region of the brain in the back where the vision area is. Now, the packaging of this is done in a non-conventional way, because the other devices I showed you have a metal or titanium that's completely enclosing the device. This is impossible with this type of device. It's too small, and we need the transparency for the magnetics. And so we actually mold these devices, and we have a specialized vacuum molding process, which then produces what's called a non-hermetic packaging, in which this silicone rubber material actually adheres to all of the internal components and produces a device that can have chronic lifetime. So now one might ask, well, where could we practically place such electrodes? And how many could we use? And what might be the expected functionality that we might obtain? So this has a matter of great debate on where the best places are. Unfortunately, in the human brain, there's an area that's along what's called the midline of the brain in the back, and there's a fissure. And if you pull open that fissure, inside that fissure are the neurons that actually have most of the vision processing for the primary vision cortex. And so this is highly inaccessible surgically. And other people have tried to just put disc electrodes on the surface of the brain, just along the center line here. And that produces kind of a skewed sort of hourglass distribution, because the neurons that you want are really buried inside the brain and you can't get at them. We decided that even going to this area produces surgical risk, because you have to do heavy retraction to get in. So we looked at other areas of the brain that process vision. And we considered here regions that are more vulnerable surgically in terms of danger of venous blood flow bleeding. And so we settled finally on what's called the dorsolateral surface, which, if you put your hands in the back of your head on each side, right by that little bump, which is the inion, and you go down where your neck goes in, that's the primary vision cortex. And so this is where we're talking about putting our little collection device, our little cell phone network that's going to be in the brain, that we can call up each electrode individually and tell it what to do. So here you see sort of a diagram of where these might be. And based upon the best information that we have, this in a field of vision, you would get approximately 1 quadrant for 1/2 of the brain. And this is about where we would get the spots of light, what are called the phosphines, and that one would perceive. Now, this will produce a group of little spots of light of irregular size and irregular spacing that one then will have to use to scan the vision field in front of them and use memory to determine what you see. And I'll show you that in a moment. So in estimating functionality, if we started with an image that looked like this, we'd like to think we could do the middle image. We're probably lucky if we could do the left image. So basically, I like to say we're trying to change somebody who's totally blind into someone who's legally blind. And for someone who's totally blind, legal blindness is actually pretty good vision. So what am I talking about with this scanning and memory? Well, you would have a little collection of these dots and these phosphines, and you would have to sort of scan around. So imagine if you were looking at the world through a soda straw that had little holes in it, or if you took a kitchen colander that you used to strain noodles, and you were to hold it up and sort of move it around. Well, what happens is your memory has to build up the image in your mind in order then to perceive what's there. So this is a very, very complicated process, and nobody quite knows how it might work. Our colleagues at Johns Hopkins have done experiments with normal people in which they outfit them with headsets and they ask them to look at 4 faces, and then they can have this simulated grid of artificial vision percepts, and the person moves it around and tries to see if they can recognize the face. and they've done lots of experiments like these. And depending upon how many gray levels you have and what the spacing is and whether they're dropped out, you get a better or worse performance. Now, in case you're thinking, well, how could anybody ever use that for any vision? Well, you might be surprised. So we've done lots of experiments in our group with our colleagues at Johns Hopkins in which a normal person sits in an apparatus like this, and then they're shown where these artificial spots would be on a vision display, and then they point to where it would be, because when we stimulate in the brain, we don't know where in the vision field the person will perceive one of these phosphine spots. So what we have to do is we have to ask them, where do you see it? And then they have to point, and then we have to map them. And when we map them, we can then know where they are in their vision space. Interestingly, this is almost identical to the apparatus that was used in 1968 in the UK to do the same thing. And when you're all done, you can get a map where you know where the actual dots were because you showed it to them. And then you get the blue ones that they pointed to, and you can get pretty close. So the mapping process works. And if we stimulate an electrode, then we'll ask them, where do you see a spot? And then once you have that, then even here, these again, normal individuals who wear these complicated headsets and then actually perform visually guided tasks as though they had the artificial vision. So here you can see someone doing what's called the placing task. And their goal is to put black checkers on the white checker squares. And what they see is what's on the right-hand side. And you might think, well, how could that be useful for anything? But now and then you can see they pass a boundary, and people learn to do this. They can do it where they can get to about 11 seconds, maybe 5 seconds a square. And after about 2 to 3 weeks of training and maybe about 20 sessions, they learn actually to do it pretty well. And here, for the scientists in the audience, here you can take a look at, it doesn't even really matter how many spots you give them. It doesn't matter how many grayscale levels you have. What really matters is the amount of time that you have to learn. And you can see that they can well perform above chance. Now, an even more incredible demonstration is this mobility task, in which we have an artificial maze that is in the computer, and the person's task is to go from room to room, find the letter on the wall, identify it, and then go and find the next letter in the alphabet in the maze. What they see is on the left. And you see, we even put visual obstacles here to try to confuse them. And of course, we can rearrange the maze at will so they can't memorize anything. And remarkably, they can go through a maze of the letters of the alphabet in about 20 minutes after they train for a period of several weeks. And here again, you can see that it's really based on the number of sessions. The more sessions they do, the faster they're able to do the task. And it doesn't even really matter what the gray levels are, what the number of phosphines are. What matters is how long do they have to train. And so this gets back to the idea that your brain is a powerful vision processing machine. And if we are able to tap into that ability to learn, we're hopeful that this artificial vision, which won't be like biological vision, could be something that could actually have usefulness. So the results of this is that the subjects who had the artificial vision, they adapted to the phosphine-like images. And with extremely limited number of phosphines, they could perform simple tasks. And the performance improves with practice. It overwhelms any other variation. And these days, we might even be able to do things like have vision processing that looks at the scene and identifies something like a car and then presents to them a unique pattern of phosphines so they know it's a car. So there's a lot of possibilities for this. To understand how significant it could be, there are people who have had other brain-based vision prostheses, and their reports are quite telling. One individual had 9 phosphines, and yet that person was able to use those phosphines to better detect where the edge of the curb was. And that allowed the person to navigate to a job that was much further away from their house than they ordinarily could. So could a phosphine-based image be sufficient? You can make a pessimistic or an optimistic conclusion depending upon how you want to interpret the results. But certainly for the intracortical approach, where we go directly to the brain, if we can learn and understand the language of the brain, we can possibly do other things, because the brain is extremely good at taking information and refining it if you put it in a way that the brain understands. So we are now in a portion of this trial or this project to do a clinical trial. This has been a long road to get here. We're now funded by the NIH for this clinical trial. And we spent about 2 to 3 years doing all the regulatory testing, the safety testing, the testing of this to determine longevity, viability, sterility. And these were major challenges because our devices were so tiny. But now we're in what we call the clinical phase. And that's going to be a 3-year project in which we will get 5 volunteers. And the goal will be to assess the stability of our interface, to map out these phosphines, to figure out what we could use them for, and then to see how it could actually help their quality of life. We'll probably implant them in 3 groups, increasing the number of devices as we go along. And we are right now recruiting the subjects for the trial. And we like to call them participants because they really are. We think of them just like the first astronauts were when they went into space. No one was quite sure what they were going to encounter. Well, when we start stimulating the vision cortex, no one is quite certain what language we'll be able to discover to talk to the brain. So they really are pioneers, and they really are taking us to a new place that we haven't been scientifically before. So after years of doing testing in animals, in the laboratory, we're convinced that we have a mature design that, even though we'd like more electrodes and more channels, it's sufficient to be able to do the test. The animal psychophysics work that we did supports a decision to implant in humans, but animals can't tell you what they see, even if you devise experiments that are well designed. And so our assessments in our prior work and our plans for the human psychophysics support the decision to implant about 600, maybe 40 of the devices, each with 16 electrodes. And after 20 years of work, it's now prepared for the clinical trial, and we are now recruiting. We have a very dedicated team from 8 institutions. We have over 50 people working on the project, and every one of them are just a delight to work with. And here you see some of the faces of these people. Some of them were with the project and are no longer with the project, but all of them are just major contributors. Many of them here, IIT students and faculty members. This was at a kickoff meeting we had in 2017. And I just show you this. You can just see on the faces of everybody here the enthusiasm for the project. The support has come from a variety of agencies and foundations. IIT as a university has supported it, University of Chicago, and private donors have supported it. and they've been especially helpful because the money can be applied in ways that you can't do with federal funding. And in particular, I'd like to mention Craig Duchessois, who's been just a champion for this project. This was at a kickoff event that we had in 2017 at the University of Chicago. Some people here may recognize some faces. Walter Nathan isn't with us anymore. This is Myri Pritzker. Here's Dean DePaula, John Georgiadis from biomedical engineering. Here's my daughter, who's working on the project. So I was quite thrilled about that. But a lot of very dedicated people, and it's been a pleasure to work with them. And I consider it a privilege to work on this project. So that's the end of my presentation. And if anyone would like to ask questions, I think they have a way to manage the questions. And I hope you found this informative and that you're as excited as we are that after such a long time that Illinois Tech is now going to be the one to test this novel brain interface.
Well, thank you so much, Phil. That was absolutely fascinating. We do have some questions coming in. The first one was from Jason, I believe it's Esterhusen. He had asked a little bit about human trials. How could someone participate as a clinical subject if they were interested?
So we are recruiting right now. In fact, this Thursday, there will be a Facebook Live event in which we will be giving information about it and recruiting. So the trial has actually started from the official standpoint. We're in the recruitment phase. And so there is a email address, and it's very simple. It's icvp@iit.edu. So if you know of someone who's interested, or you yourself possibly would be interested, then you can email that address and you can get more information.
Perfect. And along those lines, we had some questions. What ages are participants in the clinical trials?
So we have what's called a inclusion-exclusion criteria, and these can actually be seen on clinicaltrials.gov where we're listed. But the inclusion criteria has that the individual has to be between the ages of 18 and 65.
So we have a question here from Peter. Do you see neuromorphic computing as having a role in coming years?
Well, it depends what you mean by neuromorphic computing. If you mean a computer that mimics what our brain does, our brains certainly aren't like digital computers. Our brains are more like what used to be analog computers, or our brains are really like digital computers in which all of the digital thresholds of all of the gates are constantly changing. So there's no doubt that as we go forward, we become biologically inspired in our technology. I don't think right now anyone even would know how to write a program, if such a thing would even exist, for a true brain neuromorphic computer. And so if you think about how our own brains get programmed, it isn't because somebody programs it into us, it's because it basically adapts as neural networks. So this is a field of enormous possibilities that we haven't even started. We're the amoebas crawling out of the slime in terms of that technology development.
So along those lines, we have a question from Marty Cooper. Hello, Marty. Thank you for joining us. If we know that it takes years for a child to map neurons, won't people be able to do their own mapping?
Yes, so in the very early stages when a child's vision system is developing, as is everywhere in the brain, what's really happening is that connections are being trimmed back. And they're being trimmed back in such a way that it satisfies the goal of whatever is being pursued. So for example, it's not that you learn to walk, it's that you learn not to fall down. And there's a big difference between those. And so the idea that you could simply take the information and just shove it in and just let the brain figure it out, that's been one that's been debated a lot of times. And one could hypothesize that if you outfitted a newborn child with a brain vision prosthesis, that then their brain would just adapt optimally to that prosthesis. There's evidence of that in cochlear implants, in which very young children now have gotten them, even infants. But certainly at the stage that we are at, testing on infants is not in the realm of possibility. So by the time you get to be of even age 4 and up, your vision system is developed and it's tuned. And the vision system is tuned in very, very specific ways. Hubel and Wiesel won the Nobel Prize for that, in discovering that. And if you try to put the information in a way that's against the tuning of the brain doesn't really know what to do with it. The idea that the brain can just adapt to anything that comes in, that isn't really true. What it can do is it can use its natural tuning in order to optimize the interpretation of what's coming in. So really, I think you've touched on a topic, which is that what we're doing here is not just about vision. It's really about an interface to the brain that, with the novel technology we have, which, by the way, Marty, is a bunch of little cell phones in your brain, okay? So it really is about how we can form this interface to the brain. Will it be stable? And then what can we do with it? And can we learn to talk the language of the brain? And we're very hopeful that despite our arrogance of how we think we're going to put information in, we'll discover something else and there'll be an aha moment. And the person who's implanted will say, oh, wait a minute, I just perceived this. And then we'll figure out we can use that as a different way of talking. I hope that answers your question.
And I think our last question, unless anyone else has any others they would like to type in the chat, is there a difference in the approach between vision loss due to injury, disease, or genetics?
Yeah, so for all vision prostheses, they are put in physically at a location in which you have to have residual neural function. So if you go to the retina, you may have lost the photoreceptors in the retina through a disease like retinitis pigmentosa, but you still have the neurons that are beneath there that you can activate, and then those signals go out the optic nerve. If you go to the optic nerve, you still have the neuronal structure of the optic nerve. If you go to the middle of the brain, the thalamus, you have that structure. When you go directly to the brain, you have to have sufficient intact neural processing capability to be able to do something. So someone who never had vision probably doesn't have the development of the vision cortex for us to tap into. And there's a lot of good evidence for this, for people who have recovered vision late in life when they didn't have it when they were infants, and they were very confused by what they perceived. Things didn't make sense to them. People who had infant cataracts, for example, and then years later they got corneal transplants. If you have disease, it depends upon what's been affected. If the vision cortex has been affected by damage or disease, it's like saying your computer's smashed, okay? And so it really depends upon where the visual defect is and what regions we're tapping into. And by the way, there's other places that are beyond just the entry point into the brain. And we're actually, in our work, we're tapping into centers that are a little bit higher than that. So there's a lot of possibilities. You got a lot of neural machinery in your brain that's dedicated to vision.
Well, thank you so much, Phil. This has been incredible and so educational, I'm sure, for everyone in the audience. Marty asked that I share a brilliant talk, and thank you so much. Um, So for everyone on the call, just a reminder that our next Alumni University session is on May 4th from 5:30 to 6:30 p.m. Dr. Ali Sinar will be joining us to talk about his research in diabetes. So thank you again, Phil, and thank you everyone for joining us.
Thank you,
Have a
everyone.
great night.